Sorry, one moment. Okay, just getting feedback there. My apologies. Okay, welcome everybody. Thank you for joining us today. Before we get started, we have some housekeeping rules to ensure that you have the best experience. First, to make sure to choose your language, please click on Interpretation in the navigation bar at the bottom of your screen and select the language you want to listen to for today's presentation. If you wish to listen in French, you will need to select Interpretation and then mute original audio. Please note that we will be recording this session in both languages for those who are unable to attend. To ask a question, please enter it into the Q&A chat on the bottom of your screen. We will do our best to get your questions answered during the Q&A sessions. Please note that MS Canada does not approve, endorse, or recommend any specific product, therapy, or service, but provides information to assist individuals in making their own health and wellness decisions. One moment, please, and we will be getting started.
Hello and welcome everybody. My name is Pamela Canellis and I'm the Assistant Vice President of Research at MS Canada. We're very pleased to be hosting today's event. Thank you for all being here today and listening to about some exciting updates from the Canadian Prospective Cohort study to understand progression in MS, also named CanProCo. CanProCo is supported by its funding partners that include MS Canada, Brain Canada Foundation, Biogen Canada, Roche Canada, and the Government of Alberta. So joining us today are the CanProCo Executive Board Committee members who are the lead researchers on the study and pillar coordinators from across the country who've been leading and supporting the study. Today, you will have an opportunity to hear an overview of the CanProCo study, as well as the 3 scientific pillars that are integral to CanProCo. And these pillars are neuro-imaging, neuro-immunology, and health outcomes epidemiology. There will also be a Q&A session following each presentation, and we encourage you to ask questions. So we're ready to get started, and I'm excited. I have the pleasure of welcoming Melanie Gannett, Lead Study Coordinator based at St. Michael's Hospital, who'll be moderating today's event. Welcome, Melanie.
Hello, everyone, and thank you again for taking the time to join us today to learn about CanProCo and what we've achieved since we last met in 2021. It's so great to know that many of our wonderful participants are here today, as well as other valued members of the MS community. As Pamela said, my name is Melanie, and I'm one of the CanProCo coordinators based out of St. Michael's. It is my pleasure to introduce you to our first speaker for today. Dr. Jiwon Oh is a neurologist and the medical director of the Barlow MS Center at St. Michael's Hospital. She is also an associate professor at the University of Toronto, where her research focuses on developing advanced MRI techniques in the spinal cord and brain. Dr. Oh is the lead investigator for the CanProCo study, and please join me in welcoming Dr. Oh, who will now give you an overview of the CanProCo study.
Thank you, Melanie, for the kind introduction, and thanks to MS Canada for assisting the CanProCo group with hosting this event. And most importantly, thank you to our participants for all that you've done to make this pretty incredible study happen. We have a number of updates for you today, and I'm just going to start by giving you an overview of CanProCo. And I know many of you are very familiar with the study, but it always hopes to kind of jog our memories in terms of why this study came together and what has been happening in the last number of years. And then later today, after my overview of general updates, you'll be hearing from many of our scientific pillar leads. And so you'll be getting a good dose of some of the more scientific aspects of what has been happening in the study. So just give me 1 second, because I'm going to share my slides and just want to make sure. Can everybody see my slides? And I think it's actually in speaker mode. Is that right? Okay. Do you see the full screen? Yes. Okay, great. All right. On behalf of the entire CanProCo group, I'm really pleased to provide you with the 2023 update of what has been happening with the CanProCo study. Again, just a bit of a reminder, because you, our participants, are the ones that are coming in yearly to make this study happen. I know you're well aware of the general background and rationale behind the study, but as a reminder again, I think we all are well aware, you as people living with MS, but we also as clinicians and scientists doing research in the field, that one of the greatest unmet needs in MS are better treatments for progression in MS. And that includes progression that we see in progressive MS subtypes, but we are getting more and more information indicating that progression is something that happens across the entire spectrum of MS, sometimes even in the earlier stages of relapsing MS. And so this is why it's so apparent that it's something that we really need to understand better. And most importantly, it's something that we need to develop better treatments for. And the only way you can develop effective treatments for something is when you have a good understanding of what causes something like progression. And so this is really the overarching objective of CanProCo. We recognize in the field of MS that in the last 2 decades, we've made an incredible amount of progress in our understanding of the disease, in developing better treatments, and in recognizing what factors are important to maximize clinical outcomes and the quality of life of people living with MS. But when it comes to progression itself, it's really clear that it's very, very complicated, and we don't yet have a great grasp of what are the various different factors that cause progression in MS. And so that's really the overarching goal of CanProCo. First of all, it's a recognition that there's a huge range of factors that cause progression in MS. And so CanProCo was specifically designed to understand how this wide spectrum of factors are associated with disease progression in MS, and to understand how these factors relate to one another to influence progression. Because again, the first step in order to develop better treatments for progression is to develop a very good and sound understanding of what causes it. And again, that really is the goal of CanProCo.
My apologies, Dr. O. I accidentally moved you backstage.
Okay.
I'm very sorry.
No problem. So sorry, everybody. I'm just going to share my screen again. Can everybody see my screen? CanProCo, the mission? Okay. Okay, so many, many years ago, and I want to emphasize that CanProCo, obviously today you have many people from the CanProCo Executive Committee, which are our scientific pillar leads, here today sharing information. But CanProCo has been a huge team effort that extends back nearly a decade. Nearly a decade ago, many investigators in the field recognized that it would be very important to design a study like CanProCo, and that's when the brainstorming started to happen, funds were raised, this group of investigators came together, and it was actually back in 2017 when many of the current CanProCo Executive Committee members came together to try to design a study. But leading up to that time in 2017, obviously it required many years of fundraising that was spearheaded by at the time, the MS Society of Canada, which is now called MS Canada. So the mission of CanProCo was to establish a team of investigators and design a comprehensive study of people living with MS in Canada. And this was again to answer important questions related to progression in MS. And the study was envisioned at the time that it would include investigators from 3 very important scientific pillars, because there was a recognition that in isolation, obviously, each of these scientific fields has contributed immensely to the field of MS, but it's when these scientific fields can come together and the investigators involved in these studies can share knowledge and skills that we're able to really make headway in terms of better understanding progression. And so the 3 scientific pillars that from the beginning were selected to be involved in CanProCo included epidemiology and health outcomes, neuro-immunology, and neuro-imaging. So this is a timeline, and I already started talking about this, but it goes back to February of 2017 when MS Canada issued a call for applications. And this is when our group of CanProCo executive committee members came together, each bringing expertise in one of those 3 scientific pillars, and we were awarded a planning grant. And for a year, we came together, it was just short of a year, 10 months, and designed what we thought, using the funds that were available, would be the best way to design a study in Canada that met the goal of trying to better understand the variety of factors that cause progression in MS. In May of 2018, we submitted a full grant application that was nearly 1,000 pages. There was an international grant review panel that came together in the summer of 2018 to review our grant. And after a day of, actually, I shouldn't say that the grant review panel reviewed the grant for a number of weeks. And then we came together with the international grant review panel and answered any queries that they had. And we're very fortunate that in July of 2018, we were awarded $6.6 million to launch CanProCo and to conduct the study over a period of 5 years. Another industry partner came into the fold in October of 2018 and contributed an additional $2 million. And then in the spring of 2019, CanProCo was launched. And then in December of 2019, the government of Alberta also contributed an additional funding of $1 million that was to be used for CanProCo related studies within the province of Alberta. Related to the COVID-19 pandemic that it seems like the world is finally emerging from, we, like many other, all other studies, I should say, around the world, had to put a complete halt on all CanProCo study activities for a minimum of 6 months. But despite this, I'm really proud to say that we were still able to hit nearly 95% of the recruitment target, and there wasn't too much more of a delay. So I think this speaks to, again, the willingness of you, our participants, of willing to be part of this study, despite the fact that there was an ongoing international pandemic. And then in February of last year, we completed recruitment, and then I'll tell you more about this later. And then in April of last year, we were also awarded an additional $1.35 million to include pediatric onset MS patients within the CanProCo infrastructure, which is very exciting because the pediatric cohort is another example of a Canadian effort that really has made a mark in the field. So as you can see, lots has happened. And today we're going to be updating you on the latest that has happened within the last year. But again, wanted to emphasize that this has now been an effort that has been ongoing for over 6 years, the study activity itself. But this was something that came together over nearly a decade and many, many different people involved. Again, MS Canada spearheading the fundraising related to this study, but this is a really unique example of a study of not only various investigators from around the entire country coming together, but also various funders coming together, which include the MS Society of Canada, Brain Canada, which is a federal funding agency, as well as our industry partners, including Biogen, as well as Roche and the government of Alberta. So to summarize, I know that you're all really familiar with this, but CanProCo is, for now, a 5-year observational study that's taking place at 5 of the largest Canadian academic research institutions that have large MS centers. Initially, we did have many different MS centers from around Canada quite interested in participating in the study, but we weren't able to include all of them. And this was related to the fact that it actually is quite expensive to run a large cohort study like this, and there were bare minimum criteria that each site needed to meet. So we did have to restrict to 5 sites only, but moving forward, we are looking at ways that we can bring more Canadian institutions with an interest in MS research into the fold. We have many investigators around the country working on this study, nearly 50. I am the project lead, and I'm based here in Toronto at St. Michael's Hospital at the University of Toronto. And then our executive committee consists of the following pillar leads, and you'll be hearing from many of them today. But as you can see, there's a number of representatives from all of the different sites representing different scientific fields, and it really has always been a group effort. And because of the dedication of our executive committee members, that we've been able to continue as a cohesive team to push this study forward. As a reminder, this is what the CanProCo study design looks like. So everybody is part of something that we call the foundation cohort, which means that you come in for yearly study visits. You have a neurological exam done. You complete many different questionnaires. You use the iPad to do different clinical tests. And then in some people who are part of what we call the sub-cohorts, so these are the deep dive sub-cohorts where if you meet certain criteria and are willing to, we also get research MRIs of the brain and spinal cord. We get blood samples as well as cerebral spinal fluid, only if it's done for clinical reasons. And then we are collecting data annually over a period of 5 years, but as you'll hear later, hoping to extend it in the next little while. So these are our inclusion criteria, and I'm not going to go over this in too much more detail because I think you're all very familiar with it, but just a note that specifically we were quite restrictive with people that we wanted to include in this study. And again, it was because we were doing our very best to design a study that we thought would help us to understand what causes progression in MS across the entire spectrum of MS. And so because of this, we included people with radiologically isolated syndrome, which are individuals who do not technically have MS, but who have MRI evidence of lesions that look very similar to what we see in MS. So you can almost think of these people as an MS prodrome. We also included people with very early relapsing remitting MS, because again, as I alluded to earlier, we're recognizing that progression is not just a phenomenon that you see later in MS or in progressive subtypes of MS, but it can be seen even in the earlier stages of MS. And so this was the reason for including a significant number of people living with relapsing remitting MS. And then we also included early primary progressive MS, because again, this is a subtype of MS that is not very common, but manifests primarily with slow progression of disease. And we thought it would be very important to include people with PPMS in the study, again, so that we can evaluate what causes progression across the entire spectrum of MS. This is a photo of our study team. On the right, we have our scientific executive committee of CanProCo, and you'll be hearing from many members of this team today. And then on the left, we have our entire study group. And this was actually back in January of 2019 when we had our CanProCo study launch meeting here in Toronto. This is a table summarizing our research staff, and these are the research team members that all of you, our participants, engage with regularly. So you'll see some familiar names here, and wanted to emphasize that our study team members, our research staff, really are the heart and soul of our CanProCo research team, because these are our study team members that you, our participants, engage with regularly and that you probably associate directly with CanProCo. This is a table just summarizing all of the different investigators involved in CanProCo, and as you can see, it really involves people from all over the country with expertise in different scientific fields. And then we have a very large team of collaborators as well, many different people internationally with a range of expertise that bring excellent skills to the group. And then finally, we also have an International Scientific Advisory Board, which consists of a number of members who have been pretty carefully selected by the CanProCo Executive Committee for the expertise that they have in various fields, as well as the fact that most of these individuals have had experience being involved with and leading large cohort studies from different parts of the world. So this is a slide, and I know many of you have seen it already, just summarizing generally what we aim to do in CanProCo. As you can see from this diagram on the right, which is just an example of various different factors, we really are doing our very best to look at a large range of factors that underlie progression in MS. And this is not just limited to biological factors that we can evaluate, say, in the blood. It's not just limited to focusing in on specific MRI measures that are related to progression. We're really trying our very best to understand everything from the biology all the way to different health systems factors that may be related to progression in MS. So health systems factors include things like where people live geographically, how far they live from clinics, what sort of access they have to different services, depending on what province you're in. And so again, this is trying to understand not just isolated factors, but many different things that is a reflection of the real world of what underlies progression when we see it in people living with MS. And this is really the power of CanProCo. There aren't many studies around the world that have captured so many different factors on so many different levels, trying to understand progression. And this is why we are so passionate about this study and so grateful that you're willing to put your time into this study, because we really are looking at a depth and breadth of different factors that we think likely are related to progression in MS. And nothing occurs in isolation, as you know, in the real world. And so I think this is a reflection of what really potentially can cause progression, and this will allow us then to develop better treatments. And by treatments, I'm not necessarily referring to only pharmacologic treatments. We're referring to many different things that can be done in daily life that likely have a substantial effect on modulating progression in MS. We also have a number of integrative and exploratory objectives, and just wanted to take a second to highlight this fact again, because again, I think this is one of the most powerful aspects of CanProCo, in that we're looking at the relationship between all of these various factors that we know from isolated different studies are related to progression. And again, I think this is part of the power of the study. And then we have embedded novel technologies, whether it's novel technologies in MRI or novel technologies in evaluating different things biologically, or novel technologies including smartphone applications or iPad-based clinical measures, to try to also see how we can use these tools to better understand progression in MS. And this is just an example of the MSPT device, the iPad device that's used to administer some clinical tests that I know many of you are familiar with. And then earlier on in the study, we did have a digital smartphone app called the Floodlight app that some patients did use. However, this app unfortunately has been phased out, but there are many other apps that are available, and we're looking to hopefully integrate one of these in the next year or two, because it likely is that some of these digital apps will be very useful in the clinic to help better monitor progression in MS. A few other things, as you know, because you're the people that have to go through the nearly 15 questionnaires that we have many, many different questionnaires where we're trying to capture data that we think are really important to better understand progression, including scales looking at quality of life, work productivity, fatigue, mood, as well as other comorbidities. And then in 2 provinces right now, BC and Alberta, we also have access to health administrative data, which is another important aspect of CanProCo that is not necessarily something that is widely available in many different clinical studies. Because of the healthcare system that we have in Canada, we do have provincial databases that allow us to look at different things like healthcare utilization, as well as postal codes and different things that allow us to look at on a larger scale things that may be relevant to progression in MS. So this is the goal. Again, we're capturing a huge range of factors, ranging all the way from biological factors, extending all the way to health administrative factors that we think are relevant to progression. And we're looking to see what the interaction is between all of these factors that causes some people living with MS to progress while others do not. And ultimately, again, the goal of all of this is to try to understand what causes progression, try to identify factors that we can modulate to prevent progression in some individuals, and ultimately is to develop better ways to treat progression and really a personalized approach for individual patients to treat their MS so that we can maximize clinical outcomes in the longer run. A couple of things about CanProCo. It is an innovative study, and it's because it's one of the few studies that has been designed specifically to try to understand progression. It is one of the first national efforts that have tried to do this, and I think we're doing it quite well. Again, we've collected a wide range of factors and are looking at the relationships between all of these factors, which is one of the most powerful aspects of the study. We've also designed the study so that we can collaborate with existing international networks, which are listed here, because I think it's really clear within the field that it's important to take advantage of existing resources so that we can make scientific discovery as efficient as possible. As I alluded to earlier, it's an example of a study that was funded by multiple sources, including MS Canada, but also federal funding agencies, the government of Alberta, as well as industry partners. And last but not least, it's a uniquely Canadian effort, which is something that we should all take great pride in. So in the last couple of minutes, I just wanted to summarize some milestones. So as you heard earlier, we officially launched in the spring of 2019. We had a total of nearly 950 participants that enrolled in the study. And again, because of the pandemic, there was a 6-month screeching halt to all study activities. But despite this, we were able to complete recruitment by February of 2022. And currently there are 841 actively enrolled participants, and we have lost about 100 participants, but this is to be expected in a large study like this. But obviously we will be doing everything that we can to retain your interest and hopefully have you continue to visit our study sites annually, because a study like this, the data and the value of it, I think, become more and more valuable as time goes on. So again, really grateful for the time that you've invested and really hope that you'll continue on with us until the study is complete. This is just a pretty busy figure, but just summarizing what our study visit progression looks like across all sites. And just want to show you that obviously nearly 950 people completed their baseline visit. Nearly 900 completed follow-up visit 1, and now we're almost at nearly half of participants having completed follow-up visit 3. So we're actually getting almost pretty close to the initial 5-year mark of the study. This is just a table summarizing our characteristics of people who are participants in CanProCo. And I don't want to go over this in too much detail, and we're actually preparing a scientific manuscript summarizing all of these things, the baseline characteristics of the cohort, but just wanted to highlight that because we are in Canada and are a relatively diverse population, this is reflected actually in our study population, in that there's been a real focus in the field of MS and many other fields in that study population should be reflective of the diversity in a particular area. And really happy to see that in CanProCo, we have quite a significant proportion of representation from other ethnicities as well, which I think again is reflective of the fact that this is a study taking place in 5 of the largest cities in Canada. So this will also be a substantial contribution to the field, because many of the existing studies have not been as diverse. And this is again something that I think is a unique contribution of CanProCo. So really quickly, and my colleagues will be telling you about some of these things in much more detail, but we've been very productive as a group. There's many scientific research grants and awards that many of our CanProCo committee members, as well as trainees within the committee member labs have been awarded, as you can see here, including many fellowship awards, PhD awards, as well as project grants. We have many scientific papers already published, as you can see here, and then a number of papers that are still cooking. But by this time next year, you'll likely see a full PowerPoint slide with all of the papers that the CanProCo group has published. And I think this is quite something, just because, again, we only completed recruitment last year, but the fact that we already have many of these scientific papers published and in highly reputable scientific journals is a reflection of the fact that this is a really, really important study and an important data set that the field has been very well received by the field. We've had many presentations at the largest international MS meetings, and as you can see, many of these presentations have been some of the most highly ranked abstracts at these major meetings, which again, I think is a reflection of how important the CanProCo study is. And then, as you can see, here are some papers, as well as some of the analyses that are being done in all of the pillars, including neuro-immunology, as well as neuro-imaging and epidemiology, and you'll be hearing in more detail about that. And then, as I mentioned earlier, last year we were delighted that we were able to get more funding to absorb the Canadian Pediatric Demyelinating Disease Cohort. Participants who had been children at that time, who are now adults, who are willing, we will be bringing into 2 different CanProCo sites so that we can try to see, get more information on people who had pediatric onset MS and how disease evolves over time, which is an illustration of 2 important Canadian efforts coming together, again, to try to contribute innovative knowledge to the field. Last but not least, just wanted to tell you that we are now in discussions with MS Canada about phase 2 of CanProCo fundraising. As I've mentioned earlier, a cohort like CanProCo, where it was specifically designed with the goal of understanding progression across the entire spectrum of MS, the value of this data set only gets more valuable as time goes on. So it is really important that we try to, of course, if you're willing, extend the duration of the cohort if possible. So we are talking right now with MS Canada about launching phase 2 of CanProCo. We would like to ideally extend the follow-up to 10 years, and we recognize that many of you may not be able to stay with us for 10 years, but if you are able to, and if we're able to obtain the funds to do this, we would be absolutely delighted. And again, I think it would be an incredible contribution to the field if we were able to have a national study like this that extended for a duration of 10 years. We're looking to, again, bring in many other Canadian sites in the data analysis portion. We are also looking to make the CanProCo data set openly available to qualified investigators around the world, but we need the funds to be able to host a data repository and a data stewardship framework to do this. And then we have many different international collaborations that have already started. So really looking forward to this next phase where funding will be dedicated to extending the duration of the cohort, but also to all of the complicated analyses that need to be done and to bring other Canadian sites into the fold. So as you can see, there has been a lot of progress that's been made, but we know that there's a lot of work to be done. We are just so grateful to all of you for being willing to be part of this study. We have a lot of things that we know we need to work on, and they're all listed here. But most importantly, we're looking forward to phase 2 of CanProCo and further extending the duration of follow-up and seeing what additional things that we can continue to contribute to the field, because as you saw, we've already made quite a bit of progress in contributing to the MS field. And again, I think that productivity will only strengthen as time goes on. So I'd just like to end by first and foremost thanking you, our study participants. Again, I know I'm obviously biased, but I think this is one of the most unique and probably important studies in the field to contribute to the field, because we know that understanding progression is probably our greatest unmet clinical and scientific need. Would like to also heartily thank our funders, MS Canada, Brain Canada, Biogen, Roche, and the province of Alberta. The CanProCo team, this is entirely a team effort, and without every one of our EBC members and research staff, the study wouldn't be possible. The Canadian Pediatric Demyelinating Disease Network that we're joining forces with, and the entire Canadian MS community. And so before we start our Q&A session, and Melanie, I'm sorry, I know I've gone over time, just wanted to let you know that this year we finally have figured out putting together some CanProCo merch or merchandise. We just wanted to we wanted each of you to have a little bit of a keepsake of the study. And so very soon at an upcoming study visit, you'll likely be hearing from your study team about these items. So we have CanProCo branded caps and CanProCo branded brain stress balls that we hope that each of you will be able to have as a keepsake. So with that, thank you very much for your attention. And I think we have a few minutes for some questions.
Thank you so much, Dr. O. That was a really comprehensive overview. We will now begin our Q&A for this part of the event. We are using a combination of questions that you, the audience, have submitted at the time of your registration, but we would also like you to feel free to submit any additional questions you have right now. And if you'd like to do that, you can enter your questions into the Q&A chat box at the bottom panel of your screen, and we will do our best to get to as many questions as possible. Okay, so my first question for you, Dr. O, is why was the RRMS subcohort designed to include individuals who were not on MS medicines? Why was this important?
So it was really for a number of reasons. Obviously, any participant in the CanProCo study is extremely important because we're collecting such a wide range of data. But particularly for RRMS, if at all possible, we wanted to zero in on individuals who had not yet had their immune system modulated by different therapies. And the reason we did this is because, again, the goal of CanProCo is to try to understand what causes progression in MS. And so we were hoping to get a blood sample and evaluate different immunologic and other factors within the blood that may allow us to understand what biological factors within individual people cause progression. And we also wanted to get MRI scans. So we get these very detailed scans of the brain and spinal cord. And we wanted to evaluate the tissue microstructure in people who had not yet had their immune systems modulated by disease-modifying therapies, which we know many of them are extremely effective at decreasing the immune response, which we know, at least in earlier stages of MS, is responsible for kind of propagating all of the different disease mechanisms. So it was really trying to get kind of the most, what's the word, in the natural state, biologically and from a microstructural standpoint, differences in individuals that we think over time may result in differences in disease courses and differences in magnitude of progression.
Great, thank you. We have a participant who said they attended what is called a relapse recovery visit. So the question is, why do these visits exist within CanProCo and why are they important?
So one thing in the field, we recognize that our clinical neurological exam, we use it a lot, but we know it's not perfect in terms of being able to really accurately detect disease progression, because as all of you know, MS is a very complicated disease from a symptomatic standpoint. And so we don't have the perfect tools, but this is why in CanProCo we're using so many different tools, including questionnaires, to try to detect progression. But our outcome measure is over an annual visit. If someone has evidence of disease progression in the neurological exam scale, this is somebody that we would think has meaningfully progressed over a year. And that exactly is what we're trying to look at during the study. And we're looking at all of these different factors that cause an individual to progress, whereas some people may not. And so the reason why a relapse recovery visit is so important is because when a person living with MS has a relapse, their neurological disability scale will be worse than before the relapse. But what we're trying to do is, we know that the natural history of a relapse is that people do recover over time. Some people recover nearly fully, however, some people do not. And so we're trying to zero in on people who, after the relapse recovers, have actually changed in terms of disability scale compared to the year before. Whereas if you can imagine, if someone was having a relapse and they came in for their study visit, automatically an investigator, without any knowledge of what has happened, would think that that individual has progressed, but it could be that it's related to the relapse. So what we're trying to do is, again, focus on people who are not within an acute relapse, who have actually had disease progression, at least based on our neurological disability scale, over a year. I hope that makes sense. It was kind of a long-winded answer.
I think it made sense. Thank you. Can you tell us, are there other studies similar to CanProCo elsewhere in the world? And if so, can you explain a little bit about the plans to collaborate with other teams and to support the open science practice?
Yes, so there are many, many different cohort studies around the world. And I know there were a lot of acronyms on some of my slides, but just one group that we already have an established collaboration with is the German Competence Network. That's the KNNMS study. So this is a cohort study that's been going on in Germany for, I think it's almost 7 or 8 years. And this cohort looked purely at relapsing MS. And so, obviously some of the data elements that they're collecting are different, and some of the imaging that they're doing and the biological analyses are different. But as you can see, there is quite a bit of overlap with CanProCo, because we also have a pretty large relapsing remitting MS sub-cohort. And so this is an example of a cohort that we really do want to collaborate with, because I think there's a lot of opportunity to increase numbers. Say if the German Competence Network identifies some sort of biological factors that they think is relevant to progression, they would obviously be able to validate it in our cohort and vice versa. So we're actually, and Melanie is well aware of this, setting up a memorandum of understanding with that cohort so that we can start some early collaborations. There's also a Scottish cohort called, I think it's the Tomorrow cohort or something like that, where they are also looking at very early relapsing MS. So these are all examples of cohorts around the world. Obviously, the data that each cohort individually collects is slightly different, but lots of opportunity for collaboration. And I think, again, really important, because in this era of big data, I think we're recognizing as scientists that it doesn't make sense to work in isolation, and we really need to try to make scientific discovery as efficient as possible. And this can be done by establishing the right collaborations around the world. So we purposely designed CanProCo to be able to collaborate with many of these different studies. And there's even studies in the US where they're looking at high efficacy treatment versus standard treatment in early relapsing MS. And this is called the Treat MS study. And we are looking to collaborate with groups like that as well. So lots of opportunity, and this will likely really meaningfully start to happen in the next year or two. So we look forward to updating you about some of those efforts.
Great. And along those lines, where can our CanProCo participants find publications? What resources do they have to see, in more granular detail what we're going over today?
So there's on the internet, there's many different resources that you can use. Academically, the most common and large database of scientific articles is housed on this search engine called PubMed. But this is not something that, unless you're in the scientific world, you may be as familiar with. The MS Society of Canada will likely assist us. I know that with certain publications, there have been tweets that MS Canada has put out, but I think it's also important that we as CanProCo study sites disseminate scientific articles when they're available to you, our participants. And so Melanie and I have been doing a lot of thinking about this, because we are, and I didn't get a chance to talk about this today, but we are really looking to increase patient and public involvement within CanProCo itself. And so as part of this, we would like our study participants to be very well informed of publications of the study. So I almost think we need to house on some sort of website or some sort of repository CanProCo scientific articles that have been published. But it's also important, I think, to have you involved in knowledge dissemination as results become available that actually may have meaningful impact on the lives of people living with MS. We will want to engage you, our study participants. So stay tuned, but I think it's a really important question and something that we need to have some sort of systematic way to let you know outside of obviously our annual patient engagement days.
Great. So we've just had a question come through the chat, and perhaps you can comment on this more generally. Will there be analyses based on participant sex, what medications they're on, and if relapses happen in relation to what treatment plan they're following? So I guess generally speaking, the clinical management profile, will we be doing specific analyses based on sex and the treatment profile of the individual participant?
Yes, so we are collecting all of that information within the CanProCo study, as you're well aware, because every year when you come in, these are things that your study team confirms. But those are really important factors that will need to be taken into account in every analysis. So likely age, sex, treatment, all of these things will be evaluated, trying to figure out, again, what causes progression in MS. And we entirely expect it to be a combination of factors, but we need to figure out what are the most influential factors. So yes, that will be something that will be looked at in great detail.
Okay, and someone has asked if you can comment on the contribution to MS progression due to relapse versus progression independent of relapse.
So really excellent question. This is why the relapse study visits are so important, and why it's so important that when you have your annual study visits, you do report if there has been any suspicion of a relapse or your neurologist has told you that there is a relapse, just because progression independent of relapse activity is emerging as probably one of the most important clinical concepts in the last couple of years. And we're recognizing, and I kind of alluded to this throughout my presentation, the fact that progression related to something outside of a relapse, we used to think that that was something that only happened in progressive types of MS or in the later stages of relapsing MS. But when studies have been done around the world in recent years, including at our site, we're recognizing that progression independent of relapse activity happens quite frequently, even at the very beginning of relapsing MS. And, you know, part of the issue with this concept is we don't have perfect tools to measure progression. And this is why it's so important to look at so many different things like fatigue and mood and quality of life and our neurological scales and the iPad-based tests. But this is one of the key things that will be looked at in our study. And again, why it's important to report relapses, why relapse visits are important, just because we are trying to tease out what is a relapse associated progression versus what is progression independent of relapse activity.
Okay, and I think we have time for one, maybe two more questions. So the next will be, talking about this progression that's happening independent of relapse. I think a lot of MS patients are wondering, what can they do to help prevent progression from happening? Aside from official medicines and things like that, what do we know about preventing from a patient standpoint that they can do in their day-to-day lives?
I think there are so many additional things that we likely will discover through studies like CanProCo, but as a general rule of thumb, I think the role of exercise and healthy living habits cannot be emphasized more in a chronic neurological disease like MS. And I mean, to be honest, this actually applies to everybody. Everybody should actually exercise more. Everybody should keep physically and mentally active as much as they can. everybody, if they can, should have healthy and thriving social networks, because these are all things that have over time, not just in MS, but many other in aging itself and in other neurological diseases, shown to be factors that are preventative of neurological decline. And preventing things like cognitive decline and depression and all of these things. So I think all of these, healthy living principles of just keeping your mind and body as physically and mentally active as possible entirely apply to MS. And the more studies that are done in this, and it's always hard to do exercise-based studies and studies looking at cognitive rehab and mindfulness, but every study that is done shows us how important these things are in daily life to try to maximize your neurological health. So these are things I would say that are just, as a general rule of thumb, something that everybody should prioritize, especially those living with chronic neurological diseases, but also, again, like I said, everybody, including, our entire research team. So for now, I think those are good rules of thumb, and hopefully some more kind of intricate data will come from studies like CanProCo.
Great, thank you so much, Dr. O. This concludes the first Q&A session. And I'd now like to take a moment to introduce you to our next speaker for today. Dr. Shannon Coland is an associate professor at the University of British Columbia, where her research focuses on developing tissue-specific imaging techniques. Her multidisciplinary team employs multimodal tools to achieve greater sensitivity and specificity in research, specifically around clinical trials of new drugs. Please join me in welcoming Dr. Coland, who will now give an overview of the CanProCo neuro-imaging pillar. Welcome, Dr. Coland.
Great, thanks so much, Melanie, and thanks everybody for your time today. I just want to check, is that showing the correct slide? Perfect, thank you. All right, so I'm going to be talking about imaging. Oops. There we go. These are my disclosures. So everybody who is living with MS is very familiar with MRI, love it or hate it. It has become absolutely essential for diagnosing and monitoring MS. And that's because we're able to use it to see these bright spots in these images. These are the lesions, the scars that give multiple sclerosis its name. And this has also been really instrumental for clinical trials for new therapies, because it can take such a long time, happily, to usually measure the clinical changes that are happening, but these lesions are appearing more often over time, so we can use this to tell if a therapy is effective. And one of the cool things about MRI, as an MRI geek, is that you can change the settings on the scanner and get out really different images. And so you can really highlight different aspects of what's going on in the brain. So you can see those middle images are really designed to be able to see the lesions. The one on the left is more to see the difference between gray and white matter. And the one on the right is after an injection of something called gadolinium. And that's how we can see which lesions are brand new or enlarging right now. And so this is super useful to be able to get as much information as we possibly can. So why isn't that enough? Why am I not willing to stop there? And we want to keep working on these advanced, more unconventional types of imaging. Well, there are still some limitations to MRI other than it being loud and claustrophobic. It's a qualitative technique. So it's like when you take a photograph and you can see certain brightnesses and certain colors, and then you turn around and you take it again. It's not quite the same because the lighting's a little bit different. Everything's a little bit set differently. So it makes it really hard to compare the images over time or between different groups. They're also not very specific for what is the biological change that's happening. So we can see these bright lesions, but we don't know for sure if that represents myelin damage or axon damage or inflammation or what combination of those things. We also know that there's more damage going on than just what's in the lesions, and we're not seeing that with these clinical exams. And finally, we'd like to have something that's a bit more clinically relevant. So these two people living with MS have the same disability score, the same EDSS score, but very different amounts of lesions. So it's not telling us enough about how someone's feeling, about how the disease might progress for different people. So that's why we're moving to these more advanced MRI measures. So the things that we know we can do with these is be more specific to different pathologies. A lot of the work that I do is looking at techniques that are specific for myelin, which obviously is important in MS. We can also look at tissue that looks normal on conventional MRI, so looking outside of the lesions, and we can see some of the changes that are happening there. And we can do these comparisons quantitatively, look at actual numbers for different pixels and see how they're changing over time or between groups. And some of the things that we're trying to develop, especially with this study, is being able to use these to track disease progression, which obviously is the big unmet need and what we're working on with CanProCo, and also to use it to measure potential therapeutic benefits for more new therapies that might be coming to market that might not be directed at lesions per se, but at the more diffuse damage that might be going on with MS. So this is some of the crazy looking images that we get out of our research scans in CanProCo. And so we do these extra scans for those that are willing to come for the extra time or able to. The first couple there, I won't go into detail, but myelin water imaging and magnetization transfer imaging, these are ways to look at myelin. We're getting more specificity for myelin, and obviously that is the primary target in MS. We want to know about that, both in the lesions themselves, but also in the surrounding tissue. We also look at things like cortical thickness. That's telling us not just about white matter, but about gray matter and how that might be thinning potentially. We have diffusion tensor imaging, which tells us about how water moves in the brain. And so that movement changes. If you have myelin damage, it now doesn't move just along the myelinated axon, it can get across it. And so we can tell things about axon damage and myelin damage. We have something called frequency shift imaging, and we're really using this to look for very specific types of lesions. So we're really digging down and saying not all lesions are the same. Some of them are very, very good for accurate diagnosis, and some of them are really, we think, we're still finding out, we think are very indicative of someone that might be at higher risk for progression, that might have a more aggressive disease course. And finally, often we don't get spinal cord imaging in the conventional clinical exams, usually because of time, and it's also quite difficult because the cord is really small and it moves around a lot. But we're able to do some pretty clever tricks to get really nice spinal cord images. And so there we're looking for lesions. We're also looking for the cord shrinking, and we're also looking for myelin changes in the spinal cord. So we're getting a ton of information out of that extra scan. So this is what some of the images look like when they first come off the scanner. So this is the extra brain MRI images that we're getting. And this was quite a challenge to put all this together because we do have 5 different sites with 3 different scanner types, but we've got really beautiful images, and that's really thanks to the hard work from the teams, our MR techs and our physicists and clinicians at each of those sites, and radiologists. We're super grateful for that. And this is the spinal cord imaging that we're getting out of all these sites as well. So we're really excited that this is coming together. That was really whirlwind. So I apologize for that, but I really wanted to move on to some of the exciting results. And I'm going to be featuring a few of the trainees that are working with us that are really the ones doing the work on pulling out these early results. So this first one, I'm featuring Lisa Lee, who's a PhD student in Toronto with Dr. Ji-Won Oh. And Lisa is looking at spinal cord in this particular study. And she got to present this as a platform presentation in the awards category for the Young Investigator Awards at ACTRIMS International Conference in February of this year, which was really exciting. So you can see everybody's excited about this, not just us. And so she's looking at spinal cord and specifically at this magnetization transfer imaging. And that's telling us about myelin. And so this, I won't go into details, but MTR will be higher when there's more myelin. And so she was looking across the cord and looking at our different groups. And from left to right, we have our controls. Then we have the RIS, as Dr. O talked about, our radiologically isolated syndrome. Then we have relapsing remitting MS and primary progressive MS. And you can see as you go down that path that there's lower and lower values for MTR. So we think that we interpret this as lower and lower amounts of myelin in the spinal cord. So as the disease becomes more progressive, we're seeing more of that myelin damage. And this is regardless of lesions. This is just what's going on in the rest of the tissue. So Lisa looked even closer, and she divided this up into different regions of interest within the cord. So in the middle, we have the gray matter, it makes that butterfly shape. And we have the different tracks along the outside in the white matter that run the information up and down the cord. And she looked at each of these to see if we see the same pattern. And indeed we do. I mean, pretty much all of them show that same pattern of getting worse as you go to more progressive disease. You don't see it as much in the gray matter, which is the third one. And that's not surprising because there's not as much myelin in the gray matter, so it doesn't change as dramatically over time. You see it particularly in the lateral funiculi, which makes sense to me with my simplistic physicist point of view, because that's where we have the tracks that are important for voluntary motor function, which obviously is a major factor as the disease becomes progressive, and pain sensations. So this is hopefully telling us something that we can look at beyond just lesions to know more about what's happening with the disease as it becomes more progressive. This is work by Connor Keane, who's a medical student who was working with us here at UBC over the last little while. And this was work presented at the same conference, ACTRIMS, in February in San Diego. And so here he was looking at how this myelin damage might relate to some of the clinical tests. And so this one in particular, he was looking at myelin damage in the corpus callosum, so in the brain, in the one structure that joins the two sides of the brain. And one of the reasons that we look at that is because as a physicist, that's one of the only brain regions I really know. But it's also because we know that it is quite vulnerable to damage and is affected in MS, and it can affect everything from motor function to cognitive function. So we're really interested in it. So he found that when there was more myelin damage, that's how we measure that with the myelin heterogeneity index, a higher number there, it took longer to perform the timed walk. And that was particularly true in the progressive disease cohort, in the primary progressive group. And similarly, he looked at a 9-hole peg test. And so this is the one where you have the 9 pegs that look kind of like golf tees, and you put them in the holes and you take them back out and we time it. And same thing, we see higher amounts of myelin damage when it takes longer to perform that peg test. And so this is important because it tells us that this damage is actually relevant functionally. So it is worth looking at, and it could potentially be a good proxy for these tests that have a lot of noise in them. So we carried on this work. So Connor carried it on together with Irene Vavasor, also from UBC, and Connor was able to present this work as a platform presentation at the American Academy of Neurology in Boston, which is quite difficult to get a talk for. So very proud that that was possible. And Irene also presented it at the big MRI conference in Toronto this year, different versions of the same work. And so here again, looking at this 9-hole peg test and how long it takes, and now they're comparing all of these different groups and seeing, do we see the same thing across all these groups? And here we're seeing that the myelin heterogeneity index, as we showed before, it gets higher in the progressive group when it takes longer. We also are starting to see that in the RIS group, interestingly, but not so much in the relapsing remitting group. So we wanted to break that down a little bit because myelin heterogeneity index tells us about 2 things. It tells us about the overall myelin level, how high is it, and how patchy is it? So is it the patchiness or the level that's really affecting things? Oh, I'm sorry. So when we looked at just the patchiness of the myelin within the corpus callosum, we found that that was the main factor for the relapsing remitting MS. It was more patchy when it was taking a bit longer to do the 9-hole peg test. Whereas for the primary progressive group and the RIS group, it was more that the overall level was lower when it was taking longer to do the 9-hole peg test. So this is, again, just ways of trying to find out what's happening with the myelin through these different stages of disease. And can we maybe predict if someone's going to do better or worse if we're starting to see more of this damage earlier on. So in that vein, the last little bit of work that I wanted to present, and this is very hot off the presses, Polyanka Johnson is a PhD student here at UBC working with myself and Dr. Chabolsi. And so she is looking at over 2 years now, because we have that much data, for a subset of our group. And what she did is divide the group into people that showed signs of progression over 2 years and those that didn't. And this is an early analysis. So we had about 160 people in this analysis, and about 100 of them did not show any signs of progression, and about 60 did clinically. So she divided them into those groups and then looked at our MRI measures. And so first she looked at 2 measures that tell us about myelin damage, myelin heterogeneity index, radial diffusivity. And so both of these should be higher if there's more myelin damage. And it's a little bit higher in the group that showed progression, but it wasn't statistically significant. They're very, very close between the groups. But when she looked at our MRI measures that we think have more to do with myelin or axon damage, so now the myelin's been damaged for a while and now the actual axon is damaged, then she found quite clearly that our signs were showing more damage in the progressive group than in the group that hadn't progressed over the 2 years. And so for this relatively small group out of our amazingly large cohort, this is really exciting. And this is only looking at one aspect of our study. So this is going to be extremely exciting as we can add more and more different measures to understand what leads into people, if we can predict from that baseline image how someone might be doing over 2 years and what we can do about it. So that's where I wanted to stop. I really wanted to thank so much all of the research participants. Obviously can't do this without you, and it's amazing the time and effort that goes into this, as well as from families and carers, all of the team, and also to the trainees who are putting all of the work into analyzing and understanding this data. So thank you very much.
Thank you so much, Dr. Koland. We will now begin the Q&A session for the neuro-imaging pillar. So as a reminder, we're using questions that you, the audience, have submitted ahead of time, along with any additional questions that come to mind right now. So if you have additional questions, pop them into the chat box at the bottom of your screen, and we will do our best to get to as many of them as we can. Okay, so let's start off with kind of an easy one. Dr. Colin, can you tell us, we hear a lot about gadolinium and it kind of tells us if lesions are newer versus older, but is it really that black and white? Is it just new or old lesions? How much more can we get with an MRI scan in 2023?
Absolutely, thanks. And that's definitely up my alley. So we've used lesions as a marker for a long time. I mean, that was one of the first things that we could see with MRI. So that was super exciting, but it's only that little bit of information. And as you said, we can use gadolinium, which of course means an injection, and that's not necessarily desirable to find out if something is newer enhancing, but that isn't the whole story. We have now found out that there's different subtypes of lesions. Most lesions kind of appear and they get a little bit bigger, and then they kind of shrink down, and some of them will completely go away. Other ones will persist. There's also a subset that will come and just slowly continue to grow. And so we're starting to be able to detect these with all of these different advanced measures. And we're really just kind of at the exciting early stages of understanding what that means and what we can do with that information. So with these scans that we're getting now, it's incredibly helpful because we have all these little bits of information where someone scanned 10 people here and someone scanned 20 people here. And it's not enough because, as everyone here knows, MS is incredibly heterogeneous between people. So to be able to take all of this information together and really understand it, I think is going to be absolutely tremendous. But the short answer is right now, with things like the susceptibility weighted imaging, we can detect lesions that have a vein through them, and that's really great for diagnosis, for specificity, for MS. And we can detect lesions that are slowly growing, whether that's because we can see the iron that's around the edge of it, or we can look over time and see that it's enhancing a little bit. And that seems to be telling us that there's some ongoing inflammation that might be causing a potentially worse disease course.
Okay, so this is actually the first time I heard that lesions can in fact disappear. So I've learned something new. So can you tell us a little bit about, what lesion repair might entail and why some lesions may repair themselves, whereas others may continue to kind of grow and expand with time? Is it different mechanisms or?
It's a terrific question, and I don't think anyone can answer it, but definitely Dr. Yang, who's going to speak later, will be able to answer it better than me. I think understanding the biology and the immunology behind what's happening in these different lesions is an absolutely critical goal and something that a lot of the people in the study are working on, and bringing together the MRI so we can actually see which ones are doing this, and then the immunology of the actual biological changes that we can detect at the same time will be critical for understanding that, because obviously the goal is to stop any of them from continuing on and to make them repair. We do know that it does seem that some people, if we can see a few of these expanding lesions, it does seem to mean that things are a little bit different throughout the brain. So there's something a little bit different in terms of the whole immune system probably there, not just that particular lesion. But it might have to do with, when it came on, how long the relapse went on for, what treatment. So I think there's a lot of factors.
Okay. We talk about MRI protocols having so many sequences, and we have special sequences in CanProCo that are not part of the everyday medical scan that someone would have prescribed through their doctor. If you had to pick the most relevant sequence that is standard of care, and the one that you think is most likely to become standard of care, what would you say and why?
Yeah, I mean, the most critical scan that is already part of standard of care is called the FLAIR. And so this is the one that we think does probably the best job overall of seeing lesions. So this is really critical for diagnosis for sure. And if we're seeing big changes in numbers of lesions, then that's something that I think a neurologist would be concerned about and would potentially propose some changes. And it's been a long time to get to that stage. It wasn't one of the original scans that was part of the protocol. So the fact that that has become now standard of care shows that we can change. There is always a lot of resistance because we get into a routine. Radiologists are used to seeing certain types of scans. It's expensive to add extra scans. But I'm very excited that over the last couple of years, a few other new sequences have become more commonly used. So that's really useful. So I'm actually going to cheat and say two. So one is just getting a really high resolution, what's called a T1 weighted image, so that we can actually look at the volumes of the brain. And that's super useful because if the volumes are changing more quickly, so if we're losing more brain tissue over time than we expect with normal aging, then that's something that needs intervention right away. And it also really allows us to combine that with other metrics and look back at past scans and that, if we can have that at all time points. So that'd be the number one thing that I would include, and we have started seeing it. Took a while to convince sites to include it, but now we're having other sites come to us and say, hey, we've heard this is really useful. How do we implement this and do this too? And the other one is probably the susceptibility weighted imaging, where we can look at these specific types of lesions. And Dr. O is very much an expert here, looking at these central vein lesions, which are great for being very specific for MS if you're excluding other diagnoses, as well as these what are called pearls, paramagnetic rim lesions, which means that there's an iron rim that's indicating that this lesion is continuing to grow, that we think is maybe telling us something about a different state of the disease. And it's a very fast scan. So that's attractive, and it can be done on all the different scanner manufacturers. So we're seeing that being picked up more and more. So I anticipate that being standard in the next few years for sure.
So you've talked a lot about these cool acronyms like the pearls and so the SEL, slowly evolving lesions. Now, can you tell us a little bit about the role of MRI in MS diagnosis, and whether you think it will take on a larger role in diagnosing as time goes on, as these, the physics of the MRI improve and things like this? And do you think it will overtake some of the older measures that we used to diagnose previously?
That's great. Yeah, I hope so. I think that the more that we can get more specific biologically, the more we are able to differentiate between different diseases and to make the diagnosis faster. So it's already an order of magnitude faster. My grandfather was diagnosed back in the 70s before there was MRI, and it took years and years to get the diagnosis because they just had to eliminate all the other possibilities and finally said, okay, yeah, it's probably MS. So, then you move to having MRI at all, but then you had to come back at multiple time points to see that it was changing over time. Now we can see if lesions are a different age within one scan often, and so that's much faster. We're finding that some of these techniques like myelin water imaging, which tells us about the myelin specifically, seem to be quite different between some of the diseases that look like MS but aren't, like NMO, Suzak syndrome, things like that. So I think that it would be lovely if that could become an option to put in right away if it was not a clear-cut case. But in my opinion, one of the more exciting things on the horizon is that the emergence of these ultra low field portable MRI scanners. And you guys have one at St. Mike's, we have one at ABC. It's very exciting. They're much cheaper, they're portable. So we can start to have these out into remote communities. They're much more comfortable. Typically, you just lie on a typical hospital bed and get the scan. And so being able to do these on those kind of scanners, I think would accelerate things much more dramatically, just being more widely available, less wait times, less expensive, less noisy, all those good things. So I think that's something to really watch for in the next 5 years.
Great. So this is a bit of a funny question. Why do some MRI pictures look so weird? How are they analyzed compared to the more typical images that look like a brain, what we've all kind of grown up thinking? How are they useful? What do you have to do to make sense out of them? And ultimately, why are they so exciting?
I love that question because that's exactly up my nerd alley. They do look weird, and I think that that's what makes them really cool. So a lot of them are just looking a little bit deeper at the data. So I'm going to do a bit of hand waving, but in typical MRI, what we do is we excite the MRI signal, and then we let it decay away. And depending on what the tissue environment is, the signal will go away more quickly or more slowly. So we excite it, and at some point we take a picture, and that tells us about the different environments. But for example, with myelin water imaging, we take a whole ton of pictures along that time period instead of just one. So we take as many as we can so we can really understand how the signal is going away. And that allows us to pick out which of the signal is coming from myelin and which is coming from the rest of the water so we have different programs that we put together that will analyze these different techniques and spit out numbers. So we usually get a map. So for that one, we'll calculate for each pixel how much of our signal is coming from myelin. And so we get out a myelin map, and then we'll go in and draw little regions of interest or somehow separate it out into different regions of the brain and find out what's happening in those regions. So there's a lot of work that goes into it. And so this is what a lot of our trainees are working on the day-to-day. And then usually if it's working really well, then we'll try to roll it out and make it open source so that other people can keep tweaking it and make all of these things as efficient as possible, as useful as possible, and shared, because I think that's the biggest thing, is that if one person again is doing it with one study of 10 people, it's interesting, but it's not revolutionary. So that's where things like this, where all of this data and all of this analysis will be available to everybody, is absolutely critical and really exciting.
Great, thank you. This is a little bit of a cross-pillar type question, but you showed pictures earlier of two individuals with the same, I think it was the same EDSS score, but their imaging looked radically different. One objectively looked much worse, and the other not so bad. So, why is it that MRI lesions don't always translate into symptoms, and why do we have populations like RIS who can have dozens and dozens of lesions and have a normal exam? Can you comment on that?
Yeah, that's great. And it's often called the clinical radiological paradox, which I always trip over, is, how can we have one person come in with so many lesions and quite well, and another person that has maybe one lesion and be really suffering? And so there's a lot of reasons for it. One is it can be the location of the lesion. So if your brain is actually amazing at being plastic, at finding another route to do things. We have a lot of redundancy, so we can use a different pathway to accomplish the same task. So if you get a lesion in one particular area that is maybe not absolutely critical, you can find another pathway to perform the same task. But for example, in spinal cord, there's not a lot of other pathways that you can take. So a spinal cord lesion typically will affect how you feel a lot more. If it's blocking the signal, it can't get through. Or optic nerve, if it's sitting right there, there's no other way for the signal to get from the eye into the brain. So that can have a huge effect as where the lesions are. It can also be that maybe these lesions are demyelinated. Maybe they have axonal damage, maybe they have some inflammation, but the myelin isn't that damaged, so that the lesion itself might not be as bad as it looks, right? You can have a scar that's terrible that you've had really cut right through your hand, and another one that is really just on the surface and it's not going to affect you as much. And then of course there's the other, there's this diffuse damage that we know is going on potentially, that might be having more to do with how someone's feeling, especially in the more progressive stages of the disease. So I think being able to combine with a lot of the biological data to find out if there's certain protein markers that are telling us, yes, there's active breakdown going on, then that gives us that extra clue that's really going to make the difference between just seeing one little aspect of what's going on in the disease.
Great. So I think we have time for one more question. A lot of questions coming in through the chat around obtaining MRI results. So I'll just tell our audience to please contact your local coordinator about your particular participation in the study. Okay. But for you, Dr. Koland, you just commented on these cross-pillar questions involving, the biological pillar. Has any work been done to date, or are you, as the MRI lead for CanProCo, are you interested at all in how COVID infection is affecting the MRI picture for people living with MS? Any kind of updates from the literature that you're aware of?
Yeah, no, that's absolutely really interesting and such a global phenomenon. But I think, some groups have had a harder time than others. And, because it is an immune system issue, then for sure we want to look at how it might be affecting in MS in particular when people are on immune therapies and that. I'm probably not the right person to answer that. For sure, it's of interest, and that's something that we want to know. We want to look at, how infection might have affected. But I think it's more than just the infection, right? So a lot of our experience was that, especially during the worst of the pandemic, people were very isolated. Typically couldn't do exercise or other things, socialization. All of these things are so important to all of our health. But as Dr. O was saying, particularly the impact of not doing these things might be felt more for someone that's living with MS. So it's a really complicated question. So I think that's something that needs to be taken carefully into account as the direct before and after. One of the nice things about the design for CanProCo is that we have people without MS as well participating, so we can look at some of those longitudinal effects as well. But yeah, I think that will be important to tease out, especially in the immunology pillar, in conjunction with some of the health data. Yeah, that's really interesting.
Okay, great. Thank you so much, Dr. Koland. Okay, everyone, so this concludes the current Q&A session. We're going to move on to our next speaker for the day. So I'd like to introduce to you Dr. Anthony Trabulsi. He is a professor and research chair of MS Canada at the University of British Columbia in Vancouver, of course. Dr. Trabulsi's research focus is on clinical trial design, the development of practice guidelines for the use of MRI in management of MS, and establishing advanced MRI imaging outcomes in MS and the treatment of something called neuromyelitis optica. Please join me in welcoming Dr. Trabulsi today, who will give an overview of the CanProCo epidemiology and health outcomes pillar. Welcome, Dr. Trabulsi.
Great, thanks Melanie, and thanks to everyone participating today. Just confirming you can see the slides okay? Great, thank you so much. So I'm a neurologist, and I've been in practice about 20 years in Vancouver, seeing predominantly people with multiple sclerosis and similar conditions such as NMO. You know, reflecting back on where we got our information to advise people or inform people with MS about what they can expect from how MS might impact on their lives was really based on information from the 70s and 80s. And I think that's quite out of date now. MS is different today than it was back then. It's the same disease, but how it's treated, how it's diagnosed, how we manage it, how you manage it I think is very different than it was 40 years ago. And so the CanProCo program, to me, is more than just trying to identify risk factors of progression. It's trying to re-understand what MS means today, what MS means to our community, our families, our society. And I think how we're approaching this is going to address a lot of those questions. And so one of the angles of that is the epidemiology pillar, which epidemiology often looks at large populations and a lot of data within those populations. We're taking it a step behind that. We're taking kind of three approaches, and I'm going to go through that now. Of course, there's a lot of smart people involved in this. Scott Patton, professor and psychiatrist and epidemiologist from Calgary, and Larry Lind, a good colleague here in Vancouver. He's in the School of Pharmaceutical Sciences, and he's also done a lot of work in epidemiology, so looking at the impact of therapies, for example, on healthcare outcomes. And then, of course, all the people doing the work are these are just some of the key people on our team doing the work, but there's more involved as well. Jacqueline Craig is a relatively new professor at UBC. She's done a lot of work in leading some of the data scientists or machine learning, artificial intelligence work around this, along with Roger Tan, who's in the also in the imaging pillar. Jamie is epidemiologist working with Scott in Calgary. Amir in Vancouver, and then Yuki, who's our research coordinator for keeping us all together and organized and helping me with my slides. Thank you, Yuki. It seems to go on its own. So we have several goals in terms of what we want to accomplish, and I'm going to kind of go through those some of those main goals now. So administrative data is what's available essentially collected in every province, and each province collects their administrative slightly differently. So what they're collecting is how the population, how people are using the healthcare system in terms of going to the hospital, what kind of diagnosis they're getting, what medications are being used, what resources, healthcare resources are being used, is all being collected on a provincial level and kept within the province quite tightly. And so we've there's been a lot of some previous work looking at this level of information to try to understand how a condition such as multiple sclerosis in a population such as British Columbia or Alberta, what kind of resources are being used by people with MS? Are they going to the hospital more often than other people? Some really neat work that came out of Winnipeg doing similar analysis found that people with MS before they got their diagnosis of MS seemed to be accessing healthcare more frequently. It was called the prodrome, the MS prodrome. And so that's kind of the power of administrative data, is you might be able to find on a large population level little hints of something going on that might lead to further research to understand what is going on. And does that give us a chance to intervene earlier? Does that give us a chance to find the cause of a condition such as MS? So using big data can be extremely helpful to understand a condition like MS, to look at how MS is related to other conditions such as cancer, for example, and to look at how, what kind of resources are required for the MS community. Where big data or administrative data has its limitations is it doesn't have the fine details on an individual level of, who might have progressive MS, what their level of function is. So that type of granular information is missing in administrative data, but it's still a very rich source of information. I'll give you some examples of that in a moment. One important point is it's incredibly controlled, anonymized. There's virtually impossible to identify an individual within administrative data, and in fact, it took us about 2 years of paperwork just to ensure the province regulators that we are following all the rules in place to protect data privacy and data control. So it's very, very controlled data. So here's just one example, some work Jamie's in Calgary was doing. So Jamie was looking at the MS population in Alberta and looking at rates of cancer. So there's a registry with rates of cancer throughout in each province. And Jamie was able to look at all the people diagnosed with MS in Alberta and to see if there was any differences in the rates of cancer compared to the general population, and also in people with MS not on treatment, on disease-modifying treatment ever, versus those on some type of disease-modifying therapy. And so with our first look, the good news is we're not seeing an increased risk of cancer in the MS population, which is great, and no clear increased risk with the use of some of our MS therapies. And that's really important because we're finding more and more people are still on therapies at older age, and older age becomes a risk factor for cancer. And some of the newer therapies we're using for MS are more powerful on the immune system. So what we really want to explore this even further and look at it with our BC administrative data, just to confirm and kind of expand on those findings so that we can know if there's any safety concerns as we continue to use therapies as people get older. Another important aspect that you can do with administrative data is look at the cost. How does what's the impact of multiple sclerosis on the cost of healthcare, whether it's through hospital utilization, going to clinics, or the cost of medications. And so this is some preliminary work from Amir here in BC. And so we can, now we can create numbers of that estimates the cost of MS in Canada. And that's going to be important as we, in many ways, as we try to inform government that MS is important, and it does cost society, and if we do, this type of intervention, whether it's a medication or a non-medication, such as improving exercise, improving diet, I know that's come up quite a bit. This is how we might impact the cost. And so governments really need hard numbers sometimes, as well as good hard stories, to influence policy. And so I think we're going to really contribute with looking at healthcare costs, for example, in terms of helping MS Canada and other advocacy groups improve access of care and therapies for people with MS. And this is just some of the preliminary work. So that's looking at it. So that was kind of goal one, is using all the information that's out there on a large population level. That's the administrative data. But then we wanted to look at what's missing in that information, what's missing in that data, what's missing in the old studies, natural history studies of MS, which are really based mostly on the neurologic examination that was calculated as a EDSS. And that was a very limited evaluation of how MS could affect an individual. And that's where we tried to incorporate the state-of-the-art quality of life, fatigue impact, a whole variety of questionnaires. And we had such a long list that we wanted to do, but we know there's questionnaire fatigue with any study, and we didn't want to lose people because we bored them too much with too many questionnaires or exhausted them. And so we narrowed it down to what we thought were some of the key questions that we could ask in a reasonable time frame, and that would allow us to collaborate and share with other investigators doing other studies around the world. And that's why we came down on the right panel. You can see the CanProCo questionnaires. Some of the unique things we're looking at, for example, is work productivity. How does MS affect ability to work or even productivity within the home? And so that's like a really unique research skill that Larry Lynn's group has been working on. And so with this, looking at a more detailed, granular level of how MS is impacting individuals beyond what we've been doing for the past 40 years, we felt was really important to understand MS today and in the future. And here's just some example of what we've learned from it. So this was some of those questionnaires looking at productivity, and a lot of details there, but really one of the key findings was one of the drivers in multiple sclerosis that seems to impact productivity at work or at home is fatigue. And that's not going to be a surprise to anybody, but now we've got data, we've got numbers, we've got facts, and that could lead to workplace interventions where we can say, look, if the workplace was improved to accommodate and manage fatigue, there could be savings for the workplace, because people need facts and numbers sometimes to change their workplace environment. And so we're hoping that looking at MS from a much broader scope in the study, we can get the information we need to understand MS, but also to improve workplace environment, for example, or even home environment. And I've always found in my practice that fatigue comes up quite often, but also misunderstanding from people in your lives what fatigue means and why you look well, but you don't feel well. And it's such a challenging conversations in families and workplace. And now we'll have some data to support that conversation. And that's great work by Elizabeth, one of our researchers at UBC. So the third aspect, so we've got the first aspect, which is using all the information in the province available, but it doesn't have the fine details of what's happening on the individual level. The second aspect was looking more deeply at individuals with MS, collecting information we haven't collected before, can't get from administrative data. And then the third aspect is, well, let's tie this all together. Let's tie it into the immunology pillar, which you'll hear about next, the imaging. Let's see, and that's where we can use machine learning, which is essentially a computer statistical technology that can take a lot of complex data and start looking for patterns in that data that might allow us to better understand the interplay between immunology, imaging, and clinical outcomes, and then how that can identify people who are early on people who might be at risk for progression, but also identify people who are doing quite well. And I think that's going to be really important in clinical practice in terms of counseling. And then with all of that, what we hope to do is not stop there, but again, turn this into personalized medicine. In other words, using all the information we can have on an individual from what we've learned from this study to say, okay, this is as good as our crystal ball is today, and these are some considerations in terms of planning for medications, lifestyle, what impact could, for example, exercise might have to improve your long-term future, for example. And then we can also use this to help advocate for more resources for the MS community in terms of healthcare resources, social supports, and research support. So we've got a lot of dreams that we want to do with this data, but I think we're going to achieve a lot. And what's really unique about the CanProCo study and well, besides him, the wonderful leadership of G1O, is this ability to not just look at epidemiology or just look at MRI, but to look at it all integrated together along with the immunology. And I think that's incredibly unique at this, to do in such a big scope like this. So thank you. That's kind of all I really, a quick overview of what we're trying to accomplish in the epidemiology pillar.
Thank you so much, Dr. Trabulsi. Very informative. We are now going to move on to the next Q&A session with this time with Dr. Trabulsi. So a reminder, we have pre-submitted questions. If you have new questions that have come up as you've listened to Dr. Trabulsi speak, please put them into the Q&A icon there at the bottom of your screen, and we will do our best to get to as many of them as possible. Okay, so I know you may have gone over this during your presentation, but Dr. Trabulsi, what are some examples of administrative data people might be surprised are collected? Like we think about, going to the doctor or filling prescriptions, but a lot of this is collected and we don't really know about these vast repositories. So what might someone be kind of surprised to know is there?
Yeah. So a lot of it is, is really quite simple things like your diagnosis. So whenever you've gone to the emergency department or family doctor, whenever we always have to enter what the diagnosis of that visit was for. And so that's how we can kind of identify, for example, people who would have multiple sclerosis in that type of data without knowing their names, of course, because again, everything's very anonymized how we work with that data. Other things would be medication utilization is probably one of the key things. And some of the ability to collect the stuff is relatively recent and varies province by province. Each province has taken different approaches to centralizing and collecting all of that data. Some other things that could be collected, I mentioned, was the cancer registries. So being able to and I think that's really important to understand the relationship between cancer and other medical conditions, infections. So a lot of useful information's in there. What probably isn't captured as much might be some of the outpatient care. For example, with multiple sclerosis, we often give, for relapses, intravenous steroids at the clinic as opposed to in a hospital. And so that could be a potential gap. And so part of working with administrative data is trying to understand how complete it is and where the gaps might be so that we're not making conclusions based on incomplete information. There's always going to be a bit of incomplete information, but we want to, have to be, a lot of what we do with that data is try to understand how complete it is and what it's truly representing and where the gaps might be.
Great, thank you. So talking about a person's right to privacy and how that works with research, can you tell us who has the right to access these administrative data and how are individual privacy respected in the context of research, and especially as it relates to using administrative data and something like CanProCo, which is a study in which people actively enrolled?
I guess with the the administrative data, I guess it's probably by provincial law that information has to be collected, and then there are laws in place in each province in terms of who can access that data and how they can access it. So they have to be, a qualified researcher. They have to pass all these privacy courses and certifications. And then they have to really clearly state how they intend to use the information so that it's appropriate. And that whole process takes at least 2 years. When that information is released to the researcher, there can be absolutely no identifying data on it. no birth dates, for example, no SIN numbers, nothing like that, or healthcare numbers could be on it. The other two is, sometimes we want to look at regional differences. Is someone, who's living in a city getting different access to care than someone living in a remote area? And that to me is a really important question. But if there's the limit, if there's only a small number of people in a remote area with the diagnosis, we can't use that data. We can't access that data because that might identify someone. So the privacy concerns are incredibly important, and I thought really well thought out for that. And likewise, when we go to the CanProCo study, everything we do with the data, with the information we collect from individuals, is de-identified. We cannot, we cannot work with research information where we can identify people. It has to be all anonymous to protect people's privacy. And research ethics boards take that very seriously. And all of us researchers have to, again, pass these ethics and privacy courses to be able to work with the data.
Right, and along that line, with privacy being such a high concern, but with CanProCo really wanting to become a shareable data set, how are administrative data treated differently? My assumption is province-specific administrative data cannot be shared in the same, even though it's de-identified, it cannot be shared in the same way as CanProCo specific data. Is that correct, or am I?
No, you're correct, Melanie, that, for example, we even within CanProCo, which is across the country, we can only, for example, access administrative data in BC as long as we have a BC researcher working with the data within BC. And likewise in Alberta, it has to be an Alberta researcher accessing the data. So, and we can't necessarily combine those data sets. We have to kind of do it in a very indirect way. And so, if we wanted to look at the same questions in Ontario or Quebec, we would need to get a researcher there and start that whole process of data access. So every province really strictly regulates access to their administrative data and doesn't allow it to leave the province.
Okay, so we've just had a question come through the chat. Do any of the data registries capture information from insurance companies like physiotherapy, massage, bracing, etc.? And would you ever consider asking patients to allow this information to be shared? So I know we collect some of this in one of our questionnaires, but from a broader sense, what do you make of that?
Yeah, no, thanks for that question. And I think it is a really important, again, that's what I was referring to, some things are missing, and that would be missing from this. I don't know how we could access that. I think our workaround is really going into CanProCo, right, where we can get more detailed data on a smaller number of people, not the whole MS population, but it's trying to combine the strengths of both approaches, as opposed to just taking one approach. So one approach would be just looking at the CanProCo study alone, just people participating and not collecting any information but the rest of the MS community. What administrative data allows us to do is collect a bunch of information, not as fine detailed as we'd like, but a bunch of information that is more representative of the whole community. And then we can use the CanProCo information, which is much more detailed, to estimate what's going on in the broader MS community with limitations of that. But I think that's what I really like about the studies. We're taking not one but both approaches and trying to marry them together to better understand MS.
Now, we like to joke around here that CanProCo has a lot of questionnaires. If you can think back, to 2018 when we were all trying to narrow down the list, what would you say was the main driving factor that led us to choose the questionnaires we did? And can you think of any that were on the table but ultimately didn't make the cut, and why?
Yeah, no. So some of the driving factors were what we thought were really important questions that we were getting from the MS community. MS Canada had done a survey of the MS community. What were some of the priority questions of folks in the community had that they would like a study like CanProCo to address? And some of the questions that, some of the key things that came up were, understanding cognitive impact of MS, fatigue, impact on families, work productivity. And so we took that community input to help choose some of the questionnaires that were used. We couldn't quite capture everything people wanted. I know diet's come up quite a bit, and I'm very interested in diet as well. It's really it was just one we couldn't take a bite out of, so to speak. But I'd hate to, but I don't want people to think diets as a topic is not important. This whole gut microbiome topic is really still a hot topic. It's just one of those things where you want to do everything, but at the same token, if you try to do everything, you're going to exhaust people, and our attrition rate would be even higher. So it's trying to find that balance of what were questions we think important questions that we could address within 5 years. One unique aspect we were really keen on was looking at work productivity. And we thought that would have a really important impact in terms of understanding MS in a Canadian context, and then help people with the knowledge how to manage their workplace or their home environment with this new data, new knowledge.
Great, thank you. Okay, we've had an interesting question come through, and it reads, I know the questionnaires ask about drinking alcohol, smoking cannabis, but I don't recall any questions about other substances, previous and/or current use. So we don't capture this, but I would be curious to know your thoughts on that for potential changes in the future.
No, I don't. I guess there's always going to be some things that are going to be missed with any kind of a study, and I guess what we can learn from our experience over this first 3 to 5 years is, well, what are we missing? And so questions and input like from these sorts of sessions brings up questions like that, so that when we go into the next phase, which I do hope we go into, we can build on what we're collecting, because it's hard to go back in time, but you can always go forward in time with new information. Again, it's that balance, isn't it, of what seems to be useful information, what we think is useful information, versus what can we accomplish in a reasonable time frame without completely exhausting people with all the questions we ask. Part of the question, back to the cannabis and smoking, for example, we also wanted things that would we could compare against data that's collected, for example, by Stats Canada or other agencies, so that, again, if we wanted to see, well, what's going on in the MS community relative to the general Canadian population or what have you, we now, we're having data points or data information that overlaps with other things being done in Canada. We thought that would be really important.
I think we have time for a couple of more questions. Maybe not using a CanProCo example specifically, but can you take us through maybe an example from another disease state or another project where there's been translation of administrative data analysis into tangible change for a person living with the disease state being studied at all, just so we can get a sense of what that might look like down the road?
Yeah, no, thank you. And that's particularly why I was really excited when Larry Lind joined the project. So he's a professor of pharmaceutical sciences, originally a Saskatchewan boy. And he's done some beautiful work with lung disease, so asthma, COPD, and also with diabetes, where his research team were able to take administrative data and look at the impact of different therapies on asthma, and how that decreased hospitalizations and other healthcare outcomes. And this was by using administrative data. His group was able to answer questions that otherwise would have cost millions and millions of dollars to do a research study of drug A versus drug B, for example. And so that has directly translated into improved healthcare for people with asthma and COPD. The other neat thing with how Larry's group has developed their approach, it's meant to be essentially kind of like an open science approach where, let's say another researcher has done a study and they said, okay, if we do this, exercise or decrease smoking, this will have this positive impact on asthma, for example. Well, then Larry's team can take that data, put it into a model, and to see what would be the impact of that intervention in the whole population of people with asthma or COPD, and kind of calculate the healthcare benefit of that. So it's a really neat dynamic approach. It's not just here's our results. It's like here's our results. We turned it into a model. Bring us your data, and we can see how something new might improve healthcare for people. So that's, to me, was a really cool translation. And so when Larry showed interest in working with us in the MS world, it's like, yay, we got like a rock star here.
Yeah, that's really cool. Just one small question to kind of close out this session. We've had this come through the chat. You talked about the work being done using the cancer registry, and the participant wanted to know, because MS drugs are being developed fairly rapidly, is that current work inclusive of the newer disease modifying therapies? And how much of a delay, I guess, would you see on the results of a registry compared to when drugs are being rolled out onto the market?
Yeah, no, great question, because the risk of cancer isn't necessarily immediate when a new therapy comes to market, because cancer sometimes is a slowly evolving thing, and it might not show up till years and years later. So with the work that Jamie did and what we're doing here, it does reflect a lot of the newer therapies that have been around. So the monoclonal antibodies and the pills, which have been around for a while. So it does capture a lot of those therapies. So I think we're pretty well established from that perspective. And they've been around for at least a decade. In some form or another, and so probably would want at least, ideally at least 10 years of them being around to get good data, I would think, just estimating that. However, the other thing though is by establishing this approach, we can keep looking, let's say every few years, every 5 years, ask the question again and again, especially if there are new therapies coming to market, just to make sure we're not missing something. So by establishing the approach now, making that precedent that we can do this, then we could keep doing it again and again in the future. But it's really getting it started to begin with was the big challenge.
Yeah, great. Well, thank you so much, Dr. Trabulsi. That was a really nice overview and a very engaging Q&A session. We're going to move on to our next speaker, everyone. So I'd like to take a moment to introduce you to Dr. Wee Yong. Dr. Yong is Dr. Yong is a professor at the University of Calgary and the Canada Research Chair in Neuro-immunology. He co-leads the MS Neuro team at the University of Calgary and directs the Provincial Alberta MS Network. Dr. Yong's research interests lie in the area of neuro-immunology, neuroprotection, and CNS regeneration. Please join me in welcoming Dr. Yong, who will give us an overview of the CanProCo neuro-immunology pillar. Welcome, Dr. Yong.
Thank you. Can you hear and see me? Sorry, can you say that loud?
Yes, we can see you and hear you.
Excellent, good. Good morning, good afternoon, depending on where you are. Welcome to this session. It is my pleasure to speak on the neuro-immunology pillar, giving you an update on behalf of our lead, Dr. Alessandra Pratt, who is based at the University of Montreal. Dr. Pratt is on holidays currently, so it's my pleasure to be here. This program involves many colleagues across Canada, and I'm speaking on their behalf as well. So I thought I'll begin with this slide just to remind you of some of the factors implicated in MS. So in this schematic, I'm showing you the bloodstream, and I'm going to use a laser pointer here. The bloodstream here, in which white blood cells, which are immune cells, circulate, and there are many types of white blood cells in all of us.
I'm sorry, Dr. Young, I need to interrupt. Your slide deck is not showing.
Oh,
Could you try again, or would you like my help?
okay. Let me try that again. So
Okay.
apologies for that.
It showed briefly before you started speaking, and then
Okay, so let me redo it. Here we are.
that's the trick. You should do your slideshow.
Yes, I'm going to try to get that up.
There
Can
you
you see
go. That looks better. Thank you very
Okay,
much.
good. All right, so using a laser pointer here. So in this schematic, I'm showing you the bloodstream in which white blood cells, immune cells circulate. We have many types of white blood cells within us, many different varieties. And normally they're very important to protect us from things such as infections. Normally, these white blood cells do not enter the brain and the spinal cord in large numbers, and that's because they are kept out by a barrier that is called the blood-brain barrier. Here is our brain and spinal cord, also referred to collectively as the central nervous system. And I'm just showing you two nerve cells, and we have billions of this. One nerve cell called a neuron connecting with another down here. So they communicate through signals, and then some chemicals are released at this end to trigger signal in the second nerve cell, etc. And that then corresponds with the rest of the body. Normally, these signals are transmitted down these long nerve fibers, also called axons. And in order to speed up the transmission process, we have these myelin wrappings, sausage-like wrappings made by this cell type called oligodendrocyte. In multiple sclerosis, the oligodendrocytes and myelin are destroyed, and so this then affects the speed of transmission of impulses, and depending on where the injury is occurring, that would dictate which part of the body is affected. Over the course of time, though, these nerve cells also degenerate as well. So the field of neuro-immunology, the pillar of which is represented here, is this interaction of cells in the immune system and cells of the nervous system. So that's an overview. Here is another schematic, this time showing you a cartoon again of white blood cells, immune cells in the bloodstream, colored differently to give you an impression that we have a variety of these, each serving different functions, that they are normally kept out of the healthy brain and spinal cord, do not enter in any appreciable extent. But in people living with multiple sclerosis, there is activation of some subsets of these immune cells. As well, some of the immune cell types are actually breaks to prevent the excessive activation of other types of immune cells, and these tend to be deficient. So in people living with multiple sclerosis, they tend to be dysregulation, if you like, of immune cells. Some subsets are overly activated, some are deficient, that are very important to control the excessive activation. So basically then we end up with overrepresentation of some what we call pro-inflammatory immune cells that then gain the capacity to cross the blood-brain barrier. And when they do, and when they accumulate in the brain and spinal cord in large numbers, they contribute to the injury. And in this example, we see these immune cells attacking the myelin as well as the nerve fiber, subsequently leading to their degeneration. So this is the neuro-immunology of MS. So one can therefore imagine a scenario in which we can collect blood from individuals living with MS and those without, or from other conditions, look into the circulation to look at the different types of immune cell types that are represented and ask if one or more of these are excessively activated or not activated or inhibited, and begin to get a picture of the immune abnormality in that individual. As well, many of these immune cells produce factors that are, or proteins that are secreted into the bloodstream. And one can therefore also imagine a scenario in which we can analyze the blood for levels of particular proteins or a spectrum of proteins. And that's what the neuroimmunology or biology pillar is doing in this CanProCo program. So here are some of our goals. We want to identify immune signatures or biomarkers in blood that could be, and I'm just trying to read the end of my slide. That could be dysregulated at the first visit in order to predict the status of that individual then and the status some time later to try to identify the basal abnormality to begin with, and to use that to predict the probability of progression, whether an individual will be at a high or low risk of progression. Through such analysis of cells in the blood, we hope also to be able to learn which immune cell subsets are most detrimental in MS and therefore how to counter them. As well, because these immune cells produce proteins that can be measured in the bloodstream, we want to measure these products so as to recognize changes that are undesirable or those that are helpful. And we want to try to link these changes of neuroimmunology to the other results that have been collected across CanProCo, including MRI, clinical status, lifestyle factors, etc. And overall, such strategy, we hope, would lead us to develop insights on how to manage MS more effectively through understanding the disease process more precisely. So here's an example of our CanProCo analysis. Just going to walk you through, beginning with the labels to your right, individuals living with MS, many of which are contributing blood at baseline and then at yearly intervals. And at each of these visits, all of these clinical MRI demographic data is captured. And at each of these visits, blood is collected. And many of you are volunteers for this, and I thank you for that. And for the blood that is being that has been collected, the aim is to look at what is called to perform a very sophisticated technique called single-cell RNA sequencing. Basically, for every cell, there is the complement of DNA that then makes RNA. And we would like to analyze the RNA within a cell and across many cells. So this is called single-cell RNA sequencing. And through such analysis, all the cells that have been collected during each visit will be analyzed for the full spectrum of RNA that that cell is producing. And because there are many cells, we therefore have data of RNA expression by a cell across thousands of cells. This will allow us to decipher which particular cell type is present and expressing RNA of a particular type that might be dysregulated or that might be inhibited in expression. So this is a very powerful technique. Basically, if you like, allowing us to look into the universe at all the stars that are in the sky and asking the content within each of those stars that are in the sky. A very powerful technique indeed. And then there are other techniques such as flow cytometry. And this protein ELISA technique basically measures the proteins that are present within the blood that has been collected. So through all of these analyses, we therefore hope to have a very good impression of the immune repertoire, the immune cells and their products existing in an individual during the time of their volunteer blood draw. This will lead us to in-depth gene-to-cell understanding of MS pathology, biomarker discovery, what can be measured and what can be used to follow the disease course, and we hope will also lead to AI prediction models of disease trajectory. Here I'm presenting to you an example of one of the analysis that is being done. So basically in this plate containing all of these wells, one well, for each of these wells, blood sample will be placed. And within each of these wells, there are already these pre-labeled magnetic beads coated with specific antibodies that are used to capture particular proteins called analytes. So you can imagine that your blood will contain a lot of proteins or analytes. When incubated within these wells, where these beads are present, if protein X is present and binds to the bead that is meant to capture protein X, we now have this scenario here. And in order to amplify the signal of detection, we go through these steps here, eventually then leading us to the identification of the proteins that are present within a given blood sample. Some results have already been collected. This is the work of Dr. Fiona Thier, a postdoctoral fellow at the University of Montreal, under the supervision of Dr. Alexander Pratt. And here she's showing us her identification of a protein called eotaxin that is steadily increased across individuals that have higher degree of disability. So basically, in this analysis, there are 225 people living with MS who have contributed their blood for this analysis. This is compared against blood sample from 25 non-MS controls. Each blood sample is subjected to analysis in this particular case for 71 proteins. And then the data is distributed according to the EDSS status of that individual at the time of the blood draw. And we see that individuals with higher EDSS score tends to have higher level of this eotaxin. Here's another example of our analysis in which many different proteins are analyzed, and one is looking at the change in EDSS from baseline to year one. And I'm not able to read my label because of a number of things that are up there, but basically the detection of this CXCL9 molecule that is associated with an increase in progression from baseline to year 1. The single cell RNA sequencing analysis is just beginning. But here's an example from an experiment performed in my lab that gives you a snapshot of the power of single cell RNA sequencing. So each of these dots here represent a single cell, and the bloodstream in this CanProCo study, we're going to be looking at thousands of cells, and each of these cells is giving us the spectrum of over 10,000 transcripts per cell. That then allows us to do analysis like this, where here are the RNA encoding different proteins, which we can then cluster into different results. This is a very complicated analysis, but it's also a very powerful one as well that will allow us to pinpoint, let's say, a cluster of cell that may be dysregulated in MS and the spectrum of RNA transcripts that cell cluster produces. So our continuing directions then is further analysis of blood samples to identify signature of severity, progression, and well-being. We're going to continue yearly collection and also analysis of blood samples to associate these immune signatures with lifestyle and MRI activity. And we will also be providing samples to colleagues who are not directly related to the CanProCo program at this point, but who may have very specialized and important techniques to help interrogate these MS specimens. So going back again to this slide that I showed you earlier on of the normal situation and of the excessive activation of immune cells that then enter into the CNS to produce injury, the neuro-immunology biology pillar seeks to identify particular immune cell subsets and the molecules that they make that may contribute to disease progression or stability or well-being. And with that, a recap of some of our goals to identify biomarkers, immune signatures, to predict how patients are going to be years from now, learn which immune cells are most detrimental, how to counter them, measure products of immune cells so as to recognize changes that are undesirable or helpful, link this to clinical status, lifestyle factors, all of which then is to develop insights on how to manage MS more effectively as a population and for a given individual. And with that, I thank you for your attention and thank the volunteers who have kindly agreed to be part of this CanProCo program. Thank you.
Thank you so much, Dr. Young. Okay, so we're heading into our last Q&A session for the event. Again, I've got some questions that were submitted by you ahead of time and some new questions that are coming through. If you have some questions, pop them into the Q&A box in your Zoom window. Okay, Dr. Young, can you tell us a little bit about what happens on an immunological level when someone starts MS medications?
So going back to the schematic that I showed you about activation of immune cells in the bloodstream, the expectation when one starts medication, and depending on the medication, is that we would be changing the status of those overactivated cells. There are some MS medications that deplete particular immune cell types, such as B cells. We have a number of treatments that deplete B cells, and we would expect, therefore, that soon after treatment initiation, compared to baseline, i.e. before treatment, that these B cells would be depleted. As well, by measuring proteins that would inform on a more pro-inflammatory state, if you like, the expectation would be that after treatment initiation, that there would be some normalization of these immune abnormalities. So this is what one would expect. And sometimes these measurements are useful to inform on efficacy. If an individual that goes on a therapy to deplete B cells does not show depletion of B cells with continued treatment, although that is rare, that can inform on the potential of that treatment potentially not going to be useful in that individual.
Okay, thank you. And what do we know so far about various prognostic or predictive factors that are already established in MS disease progression? Like, what is your team building off of, or what has your team discovered before in terms of markers that tell us what's going on and what may happen?
So there are not that many markers that are applicable across all individuals with MS, unfortunately. And I think that speaks to the heterogeneity of MS, the heterogenic cause of MS, and whether an individual is stable or have activity in the brain, or whether an individual has a normalized immune system at that time point or an exaggerated one. So the results tend not to be as clean as we would like. And so therefore, at this point, as a field, we don't really have a biomarker A or a biomarker B that we all commonly use across the board. That said, with the degeneration of nerve fibers in the brain and spinal cord, that degeneration leads to the release of what is called neurofilament into the bloodstream. And although not perfect, that is one of our better biomarker, if you like, that those with high levels of neurofilament in the bloodstream tends to have a more active degenerative process within the brain and spinal cord and tends to have a more aggressive cause of MS. We can also put a number of immune molecules together, although that is not commonly used at this point, but that can be done by pulling together a number of immune markers that inform on the activation of immune cells. We can have a picture that that individual has an exaggerated immune response at that time, and the goal would then be to normalize that. So it's a work in progress at this point. Nothing is perfect, and hopefully with the investigations that we are conducting as a group, that we can end up with a result that is going to be quite instructive for a biomarker that informs on the immune status or degeneration in the brain and spinal cord of that individual.
Great. So I think you may have touched upon this in your answer, but we've just had a question come through the chat. Are you aware of any drugs on the horizon that have different immune targets that are not currently among the targets that we use? And any thought on approaches that involve using multiple different MS medications that have different mechanisms of action? Because like you said, not everyone has the same profile of dysregulation from an immune standpoint.
Yeah, that's a great question. Many of the medications that we currently use affect immune cells in the circulation, and that's a good thing. We have medications that, as I've mentioned, deplete B cells. We also have medications that trap immune cells in the lymph node and prevent them from circulating and therefore entering into the brain and spinal cord to produce injury. We also have medications that aim to normalize the activity of immune cells in the bloodstream. But what we now recognize is that there is so-called trapped inflammation within the brain and spinal cord. So cells that have entered into the brain and continue to fester within the brain and spinal cord. And there's also a type of immune cell within the brain that I haven't told you about. That's called microglia, that is activated in people living with MS, continues to be activated, and can produce local injury within the brain and spinal cord. So thankfully, we are recognizing that as a field and now aiming to have medications that enter the central nervous system to affect these trapped inflammation. And moreover, there's also a crop of new medications on the horizon. They're called BTK inhibitors. They are in clinical trials at the moment, actually 11 phase 3 clinical trials of 4 BTK inhibitors, an amazing number of medications. Some of these trials are in those with progressive MS, and those the BTK inhibitors are exciting in my view because they enter the central nervous system and can affect the microglia that previously we have not been attending to.
Okay, I have one last very quick question for you because we're nearing the end of the event. Rapid fire question. If there was one type of analysis that you could do, but budget and resources was not an issue, one technology that you could use that excites you the most, what would it be?
Well, there is a technology out there that measures simultaneously over 2,000 proteins in the bloodstream. Our analysis that I showed you targeted over 70 proteins, but we have the capacity to look at over 2,000 proteins at the same time. I would want to do that because it would spread our horizon, beyond means that we are thinking about at this point.
Great. So we'll have to hope for that for phase 2 of CanProCo. Okay, so that concludes our Q&A session with Dr. Young. I would now like to invite back Dr. O and our other speakers from today to provide their perspectives on the future of CanProCo, what we hope to cover next time we all see you online. And then Dr. O will close the event with some closing remarks, and then we'll end the event for this year. So welcome back to our speakers. And Dr. O, I'll hand off to you.
Well, thank you, Melanie, for doing such an excellent job with moderating, and just wanted to thank all my fellow executive committee members for outstanding overviews and presentations. And I think you've really given our attendees an idea of all of the exciting work that's happening in all of the scientific pillars. I also wanted to thank MS Canada for all of their support with the study inception, logistical support, and even our patient engagement days. So thank you, and specifically to Sarah, as well as Pamela and Natalie and Shaza for all of their help. So I think Melanie posed the question, but in these last few minutes, just wanted to ask Shannon and Tony, I know there's lots that we're excited about, but if you had to pick one thing in the next few years that you're most excited about related to CanProCo, what would it be? And I'm just gonna put Shannon on the spot and ask her first.
Sure. So I think that the design for CanProCo is really brilliant in having the different pillars because we have the expertise in these different areas. So really building up on that expertise, but I'm the most excited about the next step, which is smashing those pillars to the ground and just really combining across all these different pillars. So we're getting all these fantastic preliminary results that are kind of imaging, immunology, and then the health data. And so really starting to get that crosstalk between them. I feel like as an imager, it's like saying, I don't need to go to Niagara Falls because I saw a picture of it. So we're kind of bringing in, the health data and the clinical tests as kind of your sense of touch and hearing, and then the biology is like taste and smell, although that's kind of gross, but like to get that whole power of the experience so that we can really start to dive in.
Thank you, Shannon. And then can I ask you what you're most excited about?
Yeah, similarly, or similar to Shannon, this integration of data across the pillars is exciting. Our analysis of what's happening in the bloodstream would not mean much if we did not know the clinical status or the MRI activity of given individuals. So this meshing together of data, I think, is very powerful.
Thank you. And Tony, what are your thoughts?
Yeah, so I guess when I first got involved, I was actually a little nervous about trying to predict progression, which is probably what will come out of this. It's kind of a depressing thing to predict, very depressing to predict. However, the advances that happened in the past 20 years of treatment for MS are fantastic. Like, we can really nip it in the bud for early relapsing remitting MS. And now we're seeing some benefits for progressive MS. So my vision, my dream is what we're accomplishing today in order to predict who's at risk for progression. We're going to have treatments and strategies, not just drugs, but strategies to decrease that, minimize, stop that risk of progression. So I think this CanProCo is going to be so instrumental in identifying people before they get injured and really giving them the best long-term outcome. So that's what excites me.
Thank you, Tony. And, I echo everything that my colleagues have mentioned. And I think just to add to that, one thing I'm really excited about is I think that integration piece was really nicely articulated by Shannon. But in addition to that, I'm really looking forward to having tangible messages that we can generate from this study for you, our participants. Because sometimes I think science, there's a lot of exciting science that happens, but that step to translating it to something that impacts your daily lives is always a challenge. And I think that is a piece that I'm really, really looking forward to in the coming years, because the goal is to generate knowledge that obviously has huge influence from a scientific perspective, but also in your daily lives. And I know a lot of the questions that came up today from all of you address that, and we recognize how important it is for you to have kind of tangible nuggets that you can take from the work that we are all doing together. So with that, thank you so much for your attention. Thank you again for devoting your days and hours to participating in this study. As you can see, I think we're just at the cusp of everything that we can accomplish with this study. Next year, we'll be really looking forward to telling you about what our clear plans are for CanProCo Phase 2. And in the meantime, we've already gotten excellent feedback from you today with some of the questions that you're interested in, even logistical things like how to get published scientific papers to you. So we'll be working on all of these things. And in the meantime, thank you again for your time, and we'll look forward to seeing you next year. Maybe on a virtual format, but maybe in the future we can even have local patient engagement days. So I'm wishing you all a wonderful rest of the day. Thanks for your attention, and thank you everyone for being a part of this.